mRNA technology has come from what some may have considered backwater fringe scientific discovery to being front and center in the COVID vaccine development and the incredible vaccine rollouts internationally. I do just want to take a moment to say thank you. What an incredible job anybody involved in vaccines development and the vaccines rollout have done. Incredible work. This session will explore mRNA and how it's going to change the world forever. It's my pleasure to introduce Nicola Blackwood, Chair of Genomics England, member of the Lord's Science and Technology Select Committee and Honorary Professor of Science and Public Policy at UCL, with a very special guest, Professor Drew Weissman. Over to you, Nicola.
Well, thank you so much, Vernon. And you are right, this is a hugely exciting session, and I'm delighted to be here with Professor Weissman, who, along with his colleague, Katalin Kariko, is now known as one of the parents of nucleoside-modified mRNA technology, a technology that has almost limitless potential, it seems, to express any protein and treat any disease. Now, Drew, welcome. How did you get started on this journey? What piqued your original interest in mRNA when, as Vernon was saying, really it was seen as a fringe technology?
So I moved to University of Pennsylvania in 1997. And back then, the only way to read journals was with a photocopy machine. And I met somebody who we used to essentially fight over the photocopy machine, named Katie Kariko. And I was an immunologist. She worked on RNA. We talked, we decided to start working together, and that formed a great collaboration that has lasted ever since.
So I don't think, Professor Weissman, that anyone hasn't been following this with bated breath, given the significance that it has had for our fight against COVID-19. But can you just give the audience a primer on what is different about RNA-based vaccines compared to other more conventional vaccines like the flu vaccine?
Sure. So I consider RNA to be the middleman. So the way our cells work is our DNA contains all of the information, the code for every protein that makes our cells and our bodies work. RNA is made to copy the DNA, and it copies usually one protein. And then that RNA travels to a machine called a ribosome that reads the code and makes a protein. So what we're doing with mRNA is we're delivering the middleman. It goes into a cell, the ribosome reads it and makes a protein. The ribosome makes thousands to hundreds of thousands of copies of that code. So there's a big amplification.
So, but it's true to say that when you first started working on this, it didn't go perfectly. I know that there were some big inflammatory issues. So what was the big breakthrough that took you from this being a problem in the lab to something that really had groundbreaking potential?
So Katie and I worked on this from 1998 to 2005. And we identified the inflammation. We found what receptors, what molecules were responsible for it. In 2005, we figured out how to get rid of that inflammation. And that was by changing one of the letters in the code of the RNA. That gave us an RNA that didn't induce inflammation. And at that point, we knew that this was something big, that it had enormous potential for vaccines, for therapeutics, for gene therapy, for anything just about.
And so you're starting to give away the game now, and where we're going to start taking this discussion. But my question is, can you give an explanation for us as to what makes our mRNA a truly revolutionary technology? How is this going to change the future of biotech?
So it's a bit of a complicated question. And if we look at gene therapy, currently the way gene therapy is done is you take cells out of a person's body, you infect them with a virus, and then you give the cells back. That's very expensive, very complicated. What RNA does, it's essentially a form of transient gene therapy. So you can inject an RNA and it will make protein for a couple of days to a couple of weeks, and in theory, replace that protein. It's a much simpler delivery system. It's much safer because you're not altering the DNA, and it's very effective.
So but one of the other things that it does is it's much faster. And so what we have definitely seen during this COVID period with some of the vaccines that have come forward is a much more expedited period of vaccine development compared to traditional vaccines. Can you give us an explanation of a little bit more about how an mRNA platform is so much faster and some of the ways in which your research helped with that?
Sure. So if you look at other types of vaccines, so let's say the influenza vaccine, that every year they identify what needs to be in the influenza. They then have to isolate that virus, infect chicken eggs with it, make sure the virus grows, collect that virus, purify it. That takes 6 to 9 months. With RNA, you don't need the virus. You only need the sequence. So on, I believe it was January 12th, when the sequence of coronavirus was released, and we knew the spike protein was important, we started making RNA that day. Moderna did the same. 66 days later, the RNA was in the first patients. So it's incredibly fast because you don't have to grow the virus. You don't have to make proteins. You don't have to inactivate viruses. You just get the sequence, stick it in the RNA, and you're done.
So this really is a technology that goes hand in hand with genomics, the progress and the advancement of genomic sequencing and surveillance. And if we want mRNA vaccines and technologies to advance quickly, we need to have that surveillance in place.
Oh, no, definitely. I mean, without sequencing, without surveillance, the RNA vaccines would be the same as everything else. We'd have to wait till we have the virus and then figure out the sequence of the important parts.
So could you give us a sense of how many days or weeks, for example, it would take to re-engineer a vaccine to address a new COVID-19 variant if it was necessary? I know that so far the vaccines have stood up well to variants, but there might be a time in the future where that would not be the case.
So I can tell you under GMP conditions, and what that means is that that's how vaccines are made in order to give them to people. In talking with Ugur Sahin, he said it would take them 6 weeks from the time they got a new sequence that they had a vaccine ready to give to people. It takes us a few days to make a new RNA, but there's lots of regulatory involvement in patient vaccines.
And that takes us on to the important question, I suppose, which is anxiety that people have about the pace which has accelerated because of the improvements in technology. So Moderna, Pfizer, these are the first mRNA vaccines ever to be approved for human use. And people are, they're worried about the so-called unknown side effects of mRNA vaccines. How would you respond to those fears? What would be your answer to them, given what you know about how they've been developed?
So I think what people have to understand is that RNA vaccines weren't invented last March. They've been in development for 30 years. They've been in patients. They've been given to patients for over 15 years. This is not brand new technology. I joke with some of my friends. It took a year, 10 months to make the vaccine, and people are complaining it was too fast. If it had taken 2 years or 3 years to make the vaccine, people would complain it was too slow. I think what they have to understand is that nothing was missed. No corners were cut. No signs of toxicity. The clinical trials had 30,000 plus patients in them. Every sign of toxicity was looked for and evaluated the same as any other vaccine.
And of course, you and Katerina Kariko, you were doing the underlying research into the technology for more than 15 years, even ahead of that.
Yeah, so Katie and I have been studying RNA for 23 years and studying modified RNA for over 15. So this is not new technology.
So I think that's all really reassuring, and I know that you would reassure people as well about the robustness of the clinical trials, which you have just spoken about a little bit now. But I'd like to ask you a little bit about some of the other applications and the potential future for the underlying technology, which you started to give us a glimpse of, but is a little bit what we're talking about here at CogX with how do we get the next 10 years right? And a huge priority must be infectious diseases. Would you want to give us a sense of some of the other diseases which you and your team are looking at and working on at the moment?
So we have an enormous number of vaccines that we've been developing for years. 5 of them are going into clinical trials probably the end of this year. They include diseases like influenza, a universal influenza vaccine. We're making a pan-coronavirus vaccine. And what that is, there's been 3 coronavirus epidemics in the past 20 years. You have to assume there's going to be more. So we're trying to make a vaccine that will protect against any coronavirus in a bat that has the potential to cross over into humans. We're developing vaccines for malaria and for TB, which have enormous worldwide impact. We're developing vaccines for cancer, for allergic diseases, for autoimmune diseases, for just a huge variety of potential therapeutics that I'm hoping will be made in the next few years.
But a huge priority must also be a malaria vaccine. It infected 229 million. It killed 409,000 in 2019, of whom more than half were under the age of 5. And we know that 2 Yale researchers filed a patent for a malaria vaccine using RNA. Should we be really hoping to see the eradication of malaria in our lifetime in the same way that we saw the eradication of smallpox?
Yeah, I hope so. And the issue is that some viruses are easy. Coronavirus is actually an easy virus to make a vaccine against. Malaria is very difficult. So it isn't as simple as taking a spike protein equivalent from malaria and making a vaccine. We've spent years trying to develop the best vaccines. I think we have a good shot at having an effective vaccine. We're hoping to be in clinical trials next year.
And so we're talking a lot here about illnesses and infectious diseases, where the burden of disease is largely in the developing world. And if we've learned one thing from the pandemic, it's that you need to solve for the whole pipeline, from discovery through to distribution, right up until you get that jab in the arm. Many countries in the UK and the US, they've really excelled in solving for this problem. But we've just seen world leaders at the G7 promising 1 billion doses to help low and middle income countries who are struggling to vaccinate their population. The UN says this is about 10 billion doses too short. So this challenge, it's not going anywhere. Scaling up mRNA vaccine manufacture, cold chain storage, distribution in these nations. If mRNA is going to be the future, surely those are challenges which also are going to have to be solved to get that sort of technology as a standard of care. So how can we build that sort of capacity? What do you envision being the solutions to that challenge?
So I think what people have to understand is that a year ago, RNA vaccines weren't being produced in 100 million doses a month. That they were produced at small scale in laboratories for clinical trials. So what had to happen is we had to expand production. And that's not as simple as building a bigger factory or taking Ford and telling them to make RNA vaccines. You have to build up the production of the vaccine. You have to build up the production of all of the raw materials that go in the vaccine. So all of those are what's responsible for our slowly increasing production. Once those are solved, we'll have the capacity to make large amounts of vaccines, billions of doses per year. The other thing is vaccine equity. And the vaccines currently are being made in the US and Europe, and they're mainly available to rich countries. We've got projects where we're developing GMP facilities, so sites that can produce RNA vaccines in Asia and Africa. So they have the ability to make vaccines in their own countries and distribute them locally. And I think that's going to be critical for the next pandemic or the next worldwide disease that needs rapid treatment.
And this is not just a problem for vaccines. This is also obviously a problem for therapeutics. This question extends from the question of capacity for manufacture and distribution, but also to affordability of the therapeutic or the vaccine in and of itself. And sometimes this can be incredibly challenging to resolve. As a researcher in this field, how do you feel when the product of your research is sometimes unaffordable to those who need it most?
So, I'm a basic science researcher. So my job is to develop the new therapies. But I do spend a lot of time working with pharmaceutical companies, working with the WHO, with CEPI, and with other groups, figuring out how to make vaccines on a scale and at a cost that can be delivered and afforded worldwide. The Gates Foundation says a vaccine should cost about $1.50 a dose for distribution and affordability in third world countries. That's a big jump for a brand new technology. We're working on ways of accomplishing that. And I think that's what's critical. The problem is that any new technology is expensive because it's hundreds of millions of dollars for clinical trials, and the pharmaceutical company needs to make that money back. We need to find ways, the Gates Foundation in principle, they say, yeah, pharmaceutical companies can sell to US, Europe, Japan, Korea, other countries that have a lot of money, but give us the vaccines that we can make and distribute to third world countries without your profit involved. And I think that's the best way to do this.
And what do you think about the idea, which has come out in a number of campaigns and comments about the idea of sharing the underlying technology, whether for mRNA or other, the patents to the developing world or those who struggle to afford it? Do you think that this would speed up the production and distribution? Or do you think it's actually the blockage is somewhere else in the system?
Yeah, so I think I like the idea. I think it should be discussed, but I don't think it would solve the problem. Right now, the RNA vaccines, the problem is raw materials, and there just aren't enough of these raw materials available. So if the raw materials were solved, we're building facilities in Africa, in Asia to make these vaccines. I have nothing to do with patents, but that can be discussed between the pharmaceuticals and these countries, and I'm sure they would come to a good decision.
So we've talked a lot about vaccines, preventative steps, but there is a lot of exciting work that's happening in therapeutics. We've touched on this a little bit. mRNA cancer vaccines, you mentioned that you have some work on this going on in your lab. They've been employed now in a number of trials. There are some promising results. What do you think is going to be the contribution of mRNA to the treatment or even prevention of genetic diseases like cancer?
So, cancer is an incredibly difficult subject because it isn't one disease. There are hundreds of different kinds of cancers, and each one of them is likely to respond differently to immune therapies and vaccines. We're studying particular types of cancers that we think will respond well. I think this is just technology development. Over time, we'll develop systems that work well. Those can then be expanded to other types of cancer. So I'm hopeful in the next, I don't know how many years, we'll continue to improve our treatment of cancer.
And do you have any particularly kind of promising trials or pieces of research on the horizon, or have you noted any in other labs?
So for cancer, BioNTech actually has been running a personalized cancer vaccine. And what that involves is they take the tumor from the patient and they look at all of the genes and they look for mutations. And then they make an RNA vaccine that targets those mutations. They've had better luck than other therapies. So I think this is another future direction to go for all types of cancer, which I know they're looking at.
And what about other genetic diseases? Are there targets which are being prioritized amongst other areas?
So the big one that we're working on is sickle cell anemia. And what you have to understand about sickle cell, there's an FDA approved treatment. It involves taking bone marrow out of a patient, infecting it with a lentivirus and giving it back. 200,000 people a year are born with sickle cell in sub-Saharan Africa. You can't do 200,000 bone marrow biopsies. So we've been developing an in vivo gene therapy. And what that means is that we've trained the LNPs to go to the bone marrow stem cells, the cells that make red blood cells. And we can deliver RNA that will fix the broken gene in sickle cell anemia. So we think this kind of therapy is really the future because it can treat large populations inexpensively. And we're hoping this is successful.
So I think this is something which perhaps hasn't obviously come through in the discussion and the debate about mRNA. One, I think what has come through very clearly is the potential transformational nature of the technology, the curative nature, the fact that you may be able to very quickly turn around therapeutics and vaccines. But what I think people haven't necessarily grasped is that for patients who have very grueling therapeutic regimes, this perhaps would really improve the quality of life and the therapeutic experience, which is hugely exciting and a massive breakthrough, which I don't think has necessarily had as much attention as it could have.
No, I completely agree. Everybody is focused on the vaccine, and rightly, but we're working with other people, companies to treat autoimmune diseases, which if you've got one, it is a real bane on your existence. We're working on genetic deficiencies. We're working on allergic disorders. We're working on, Katie and I joked back in 2005 that this had unlimited potential for therapies. And we started making lists and we just ran out of paper after a while because the list kept going on and on and on of all the potential diseases that RNA could potentially treat.
And the other point, of course, is that theoretically, I mean, obviously, stem cell treatment, as you described it, is very difficult to deliver in some of those countries which do not have the health infrastructure. Whereas the nature of that therapeutic, which you described, would be a lot easier to deliver, and so it also has a significant health equity element to it, assuming affordability is manageable.
No, exactly. And that's why we have funders like the Gates Foundation, who is interested in funding therapies, but isn't interested in making a profit off of it. So that gives you affordability. There's still a lot of steps to go. We are setting up GMP facilities. Our hope is that those will be used to make gene therapies for sickle cell and other diseases that affect that area.
Now, one of the things that we haven't touched on, but this is a festival of AI amongst other things, is that this is a really significant, wicked data challenge. There are many ways to write an mRNA code, to produce exactly the same protein. A number of variants is, exponential. It's really can be incredibly difficult to test experimentally. So ML, synthetic data approaches, they must be a major factor in the success of researchers.
No, that's exactly right. And I can give you two examples. So the first is we work with Betty Korber, who works at the Los Alamos supercomputer site. And what she does is she analyzes every coronavirus sequence that's ever been made. I think it's approaching 1 million right now. And through her analyses, we're identifying conserved regions between all beta coronaviruses that may allow us to make a vaccine for any beta coronavirus. The other is BioNTech, which has an incredibly sophisticated AI routine that takes the sequencing from a patient's tumor and figures out what immunogens, what antigens, RNAs to make to treat that patient. So that's critical. Without that, we're back with hammers and knives trying to figure out how to do things.
So I think what's so interesting about this technology is it's a technology which has its time because of the advances, not just in your discoveries and mRNA, but also in genomics and sequencing and surveillance, but also in the capabilities of data analytics and insights to be able to combine all those three and actually exploit this technology at the crucial moment when we needed it most.
No, you're exactly right. I mean, if any of those elements had not been developed and not available, the vaccine production would have been delayed across the board.
So I just want to ask one final question before we go to questions from the room, and that is, what other uses of for mRNA do you think there will be over the next 10 years? Because that's what we're looking at, but I suppose you could reach out further if you want, including in vivo gene therapies, because that's my personal interest. So this is my question for me and my crowd, if that's okay.
So I think the in vivo gene therapies have enormous potential. And there's hundreds of genetic diseases of bone marrow. We're learning how to target other things, so liver, brain, other organs, to treat as many different genetic diseases as possible. There's also huge advances in therapeutics. So when somebody has a heart attack, a part of their heart dies, and it gets replaced by scar tissue. Moderna, for years, has been looking at therapies delivered with RNA that can rejuvenate that tissue, that can improve heart function. That can be done to brain, to organs. So there's huge avenues of therapeutics with RNA that are now being investigated, and we're very excited about.
Well, that sounds incredibly exciting. As the daughter of a cardiologist, I can't tell you how he will want to hear all about that the minute I get off this call with you. But I have a question coming in actually from Vernon, who is cheekily getting in on the Q&A pretty quickly. And he's made an interesting point and an insight which will be relevant to our audience, which is that scientific innovation of this kind, which is so transformational, often comes from the edges. How did Drew and your team, how did you as medical scientists understand so much earlier than others the transformational potential of mRNA? I would add to that, how did you stick to it when there were so many doubters? And how can we better help others to do the same?
Yes, that's had a lot of air to it. Katie and my story about not being able to get grants, not being able to publish papers, but persevering. And we did it because when we started this, we knew that it had enormous potential. And we knew that we had to convince people. And we're both very stubborn people, so we didn't give up. And we kept fighting forward. We kept getting great results that told us, yes, this has the chance to work, and we keep needing to move forward. I think the bottom line is that we need to support basic science research. Just about every discovery, every new medical treatment is derived from basic science research. And if we don't fund that, we're going to lose out on all those potential disease cures in the future.
And also, what is notable here is the interdisciplinary elements of it, the data science plus the pure biotech plus other elements that have come in to realize this solution at the right moment. So what I'm wondering is how can we encourage that interdisciplinary work all the way through the innovation pipeline so that we can and exploit the technology once it comes into being, but also encourage the discovery at the earliest and most fundamental stage.
So I think science is much more interdisciplinary now than it was years ago. And one of the reasons that I went to Penn was because it was an incredibly collaborative university. This was 20 years ago. You would have never seen somebody in neurosurgery working with an immunologist in infectious diseases, but Penn encouraged it, and that's how Katie and I started working together. But I think that's critical because there isn't one answer to every question, and there isn't one set of hands that can solve a problem. You need input from many different fields of study.
So we will no doubt have a lot of young scientists who are starting off in their careers, many of whom will be looking at you with awe and thinking, I'm never going to get there. And they will be wanting to not only understand your path to success, but also understand, how did you navigate your way through your career? So what advice would you have for them?
So I think there's a couple of important things. The first is you have to be interested and you have to love what you're doing. You can't take anybody off the street and make them a basic science researcher. It takes certain types of personality and an interest in learning. So if that's you, if that fits your bill, then I would encourage you to explore basic science research. It's an incredibly rewarding field. It's an incredibly difficult field because getting money writing papers are not easy. But if you're inquisitive, if you're interested in disease and technology, then it's a fantastic field to be in because of the camaraderie, the friendships, the interactions with people I've all found to be wonderful.
And I've had another question in from the audience, and it's asking, are there any specific types of diseases or viruses that mRNA would not be appropriate for or just wouldn't work on?
Yeah, so we're figuring that out. I mean, we try everything that people ask us to try. I'm sure there are going to be certain pathogens, certain infectious diseases where RNA vaccines don't work well. There's going to be therapeutics where it doesn't work well, but nothing works for everything. And I'm always concerned if somebody says that this is a cure-all for every disease. I don't trust that. So yes, there are going to be things that RNA can't cure.
But it still has enormous potential. Let's just have a final little couple of questions around the ethics. So obviously, and this is a question that's come in from the audience, some adverse reactions have been reported at a low but non-negligible rate, and sometimes within hours. So is it still reasonable for you to claim that there are no signs of toxicity? That's come in from an audience member.
So the RNA has no toxicity when it's given to an animal. As far as clinical trials and post-marketing evaluations, yes, there are some unexpected, rare, I mean, a couple per million adverse events. You see that essentially with every drug and every therapy. So I never thought there would be no adverse events to RNA. What's shown up is an anaphylactoid reaction that occurs 10 to 15 minutes after the shot that's easily treated. The newest is myocarditis in young men that resolves and is easily treated. Those, we do a risk-benefit analysis. So the risk of COVID is 0.5% of people die on average. And 20 to 50% of very elderly people die. So the risk benefit is negligible.
And obviously, as you go through the process of working out the different applications that mRNA have, you will have to make choices about where to prioritize your research. We've spoken about cancer, malaria, sickle cell, infectious diseases that affect millions of people. How do you prioritize your research in order to maximize impact?
So I take a really open approach, and we don't do everything in my lab because that would be impossible. What we do is I currently collaborate with over 100 labs around the world. And the way that works is I look for an expert in the field. So I look for an expert in malaria or an expert in norovirus or in any particular disease. And I contact them and say, we have a new vaccine. Are you interested in trying this in your system? So that way we collaborate with the experts who have all the systems set up. And that allows us to study many diseases, much more than a single lab can do.
And I just have one final question, which is, you have discovered something extraordinary, which has had an impact on humanity, which will go down in history. You stuck by it when people didn't believe you and they thought you were mad. Is there anything that you would have done differently?
You know, hindsight is a great thing, and at this point, you know, obviously I can't say I would do anything different. Had RNA failed, I would have said I wish I did something else. But no, I mean, I wouldn't have changed anything in my career. We studied science, and to me, that's the most important thing.
Well, Drew, Professor Weissman, it has been the most tremendous pleasure to meet you. I think that everybody who has been in this event will have felt like they have heard an extraordinary contribution in which we got a sneak peek of what is going to come in the next 10 years in terms of discovery and contribution to ending at least a significant proportion of the pain of some of the most significant diseases across the world. And a good proportion of that will be down to the fact that when people told you to stop researching something, you said, no, I'm going to carry on. And I, for one, am incredibly grateful to you for doing that. Thank you so much. And over to you, Vernon.
Absolutely. Thank you. Thank you so much, both of you, Nicola and Drew.